This method can help answer key questions in the phylogenetic field, such as how to collect sequences and how to select the correct computational model. The main advantage of this technique is that it's very easy to learn. Generally, those who are new to this method may struggle simply because they don't know how to handle the data due to a lack of basic knowledge of the principles and methods of phylogeny. This method was first developed when we analyzed the alignment of eukaryotic sweet genes. Visual demonstration of this method is critical as there is a magnitude of choices and settings for different aspects of the alignment.
Begin this procedure with selection of 35 candidate sweet proteins from unicellular eukaryotic organisms as described in the text protocol. Align the 35 sweet sequences by inputting them into Clustal Omega. Copy and paste the protein sequences in FASTA format into the input box or upload a sequence file in FASTA format. Specify that it is an amino acid sequence by clicking the icon under the pull-down menu in the Step 1 section. Specify the output format and other parameters in the Step 2 section if necessary. For this study, set output format as Clustal without number and leave the other parameters on default settings. In most cases, the default parameters work well without any specification. Submit and run the alignment in the Step 3 section. It may take anywhere from several seconds to minutes until the alignment is finished. In the Results Summary panel, right-click the link under the Alignment in Clustal format and save the aligned sequences as 35.clustal. To open the alignment result file in Bioinformatics,